way on the side of immunology, although I'm interested in ecology and evolution.
So can you speak up? We can't hear you at all.
Sorry, I can't.
Sorry.
I'm old. I'm pleased to be here. That's what I just said. It's a beautiful campus. It's not that easy to get to from San Diego, especially on United Airlines. But somehow I made it here by midnight last night, and so my voice is a little bit shaky. So I've given this talk to hardcore immunologists many times. And it's sort of my goal to sort of proselytize a little bit different viewpoint of the immune system from the way that card-carrying immunologists usually look at it. And but I've never given this talk to such a sophisticated group of people who actually know about evolution and so on. So you'll have to forgive me a little bit. Some of the things I'm going to tell you, I know you know, but this is the talk I'm going to give you. So there is a common perception, perhaps not in this room, but elsewhere, that evolution works in a hierarchical system where complexity increases and sophistication, and it's inevitable. And let's see, here at the top, you go from plants to arthropods and vertebrates, and here at the top is a white male. That's meant to be ironic, by the way. Of course, we know that this is really not the way evolution works, but it really, and even among people who understand intellectually that this is not the way it works, there's still, all of their thinking is still colored by being a human being and looking at the universe through anthropocentric sort of eyes. And here comes from a famous immunology textbook. The adaptive immune system signaled into action by the innate immune system is the optimal host defense. And so you're saying, well, yeah, maybe that's right? But there is no optimal host defense, as I think most of you would agree. It's not as if we are eventually going to overcome host pathogen interactions evolutionarily. There will always be host pathogen interactions. And I doubt that we have achieved anything like an optimum. But this is the way people think about this. And then at least this person dealt with another issue, which is that not all animals have an adaptive immune system. In fact, only vertebrates have an adaptive immune system. And yet, most animals are not vertebrates, and they're perfectly successful. In fact, they're much more successful in terms of species diversity and abundance than vertebrates are. So how do you explain, if this is so whoops, I'm sorry. Okay, if this is an optimum, and it's necessary in order to have to defeat the world of pathogens, how is it that invertebrates are so successful? So the unspoken idea is that pathogens have in general have a very short lifespan and evolve very quickly compared to hosts. And so how could you ever hope to out-evolve a pathogen? And from the vertebrate immunologist's standpoint, the answer was that we evolved the adaptive immune system, which can evolve on a cellular scale. And so it was thought that, okay, the adaptive immune system allows you to match the evolution of a pathogen, and hence we're so successful. And yet invertebrates don't have this highly plastic adaptive immune system, and yet they are very successful. So this is the conundrum that immunologists might try to explain, and usually just do so by something like this: long-lived complex vertebrates require an acquired immune system, which implies that invertebrates are not complex and they're short-lived. So that's not going to be a very good answer to why invertebrates can be successful and not have an acquired immune system. And we all know that this is the way evolution works, not like this. It's not hierarchical. It's more like a bush. And in fact, these organisms, because they have a very short generation time, have evolved. You could argue they've evolved even more than this organism, white males. Okay, so one of the reasons that we have this perspective is that we look at the world because we're humans, and we think about all of biology as if humans are a representative. And I want to argue that there's a certain uniqueness of human disease, which is one reason why this viewpoint is a little bit warped. If you think about human disease, these are some of the diseases that you might think about. Smallpox, typhus, measles, rubella, influenza, plague, and so forth. All of these diseases have a somewhat unique characteristic, and that is that what Jared Diamond has called, they are crowd epidemic diseases. They are diseases in which they have a recent zoonotic origin. So this is an origin within the last, let's say, broadly speaking, 7,000 to 10,000 years. They require a high population density, and they are acute, and they have a high virulence. Now, what is it about human disease that's different then? And I would argue that humans uniquely completely changed their ecology about 7,000 years ago. We went from being hunter-gatherers in very small bands at low density to domesticating plants and animals and moving to high-density cities in which diseases could flourish as acute crowd epidemic diseases. And so the history of human disease ecology goes back. I mean, you can find quite a bit of information back up to about 400 BC. And at that point, these acute diseases would sweep through communities almost constantly, almost on a yearly basis, and wipe out large percentages of the population. As a matter of fact, in cities, it wasn't until the 1900s that London had a self-sustaining population. Before that, it required constant influx of individuals from rural communities because so many people died of disease in cities. So this is the type of disease that we are familiar with, and it's also the type of disease in which we've been very successful making vaccines against. In fact, all the vaccines we have are against these crowd epidemic diseases, and none of them, with an exception or two, are against co-evolved diseases that have the characteristics of persistence and latency. So if you remember back to the list, we have vaccines for almost all of those diseases. We have no vaccines for alpha, beta, gamma, herpes viruses, hepatitis viruses, tuberculosis, and so on. So there's something about this that's really quite different. And did you ever wonder why, when you were growing up, why your dogs and cats didn't have to go through the same kind of disease process? Well, maybe you're too young, but when I was young, when I was your age, when I was a young person, we all had to get measles, mumps, rubella, and everything. It was just part of childhood. These were childhood diseases, and yet, our dogs and cats were perfectly fine. Never even got a cold, as far as I could tell. But they certainly didn't have to get measles and lie in bed with pox all over them for 2 weeks. The reason is because, I think the reason is, and please, after I get finished, you can start throwing tomatoes. I think the reason is that this is uniquely human. And this this caused, as I referred to a little bit before, this caused my colleague Ajit Varki to suppose that humans actually have are selected for a hypersensitive immune system. And in fact, that's the basis, he argues that's the basis for all of these diseases such as cardiovascular disease and autoimmune disease. We've been highly selected to be able to overcome these acute childhood diseases. Okay, so this leads us, from a human-centric perspective, to have the following kinds of statements made in the beginning of immunology textbooks. Most infections are cleared. The immune system genes must evolve to keep pace with the increasingly sophisticated evasion by pathogens. And one of my favorites is, it is surprisingly still not universally accepted that any given life-threatening infection results, by definition, from an immunodeficiency, whether inherited or acquired. In other words, the only time you can really get sick is if you have some deficiency in your immune system, as if pathogens have no way of overcoming immune mechanisms. This is, ladies and gentlemen, this is the sort of culture that we have in basic immunology, and so this is why I go on the stump sometimes and try to talk about this a little bit. Okay, so here are some diseases not cleared, and I guess I don't really have to convince you that this idea that most diseases are cleared or that you have to be immunodeficient is an explanation for anything, but what you can take from this is that these are all diseases that are probably long co-evolved diseases. And I argue that this is the major form of disease evolution, of coevolution, are these long co-evolved diseases. In particular, I've been sort of reading on herpes viruses, alpha, beta, and gamma herpes viruses. And they certainly infect, there are certainly herpes viruses that infect all vertebrates, but this extends to mollusks and invertebrates as well. It hasn't been studied in all of these different species, but in terms of vertebrates at least, herpes viruses go all the way back. And so this is something that could have easily driven the evolution, or had a part in it, driving the evolution of vertebrate immunity. All right, so the reason that the invertebrates don't need, this explanation that most diseases are cleared is probably not correct. Okay, so I think I've already told you this. Immune system genes must evolve to keep pace with increasingly sophisticated evasion. This is obviously not going to ever be true, because the immune system genes cannot possibly evolve fast enough to keep pace with pathogens. This is a study some of you may have seen from Ed Holmes, studying RNA viruses. And I just want to point out two aspects of this study. This is a time series of influenza, and this is a dendrogram showing the sequences of influenza. And this is over about 30 years. And you can see how influenza just changes sequentially in response to the immune response each year in annual influenza. But even more interestingly, I think, this is the time sequence of HIV within one individual over several years. So this is why we're probably not going to be really very successful in making, as an aside, this is probably why we're not going to be successful anytime in the near future in making a vaccine to HIV. But you can see that there's no chance for a host to out-evolve a pathogen, even with the adaptive immune system that has all this fabulous plasticity. And immunologists love to use, or have begun to at least use, model systems of infectious disease. When I was learning immunology, we used things like KLH and LPS, and I spent a long time looking at the dreaded pathogen cytochrome C. And at least now, people are using model viruses. The problem is we're misusing them still. So the most common form of virus that people use right now is lymphocytic choriomeningitis virus, LCMV. And the most common form of that is a laboratory adapted version, which is called LCMV Armstrong. And LCMV Armstrong is a slightly attenuated virus that allows the mouse to make a huge cytotoxic T cell response and clear the virus easily. That's not the ecology of the LCMV. The ecology of LCMV is it's a long co-evolved arenaviruses, or long co-evolved with all sorts of murine species, and these viruses, about 5% of the mice carry them here. They carry it for life and shed it for the duration of their life. It's spread vertically, so it must be persistent, and otherwise it couldn't be spread vertically. And it also has a horizontal transmission component, but it's also persistent. The problem is that most studies are done using this LCMV Armstrong, which is a very nice model system for an acute virus infection, like a crowd epidemic disease, but it's not a very good model for the actual virus itself. Now, people also use another variant called clone 13, which is probably the more natural version of the virus, and that is a persistent virus. This virus, when it jumps to humans, as this is an aside, it causes lots of bad diseases, but it's not transmitted from human to human. There are no arena viruses that have a human as a natural host. This was an article written in 2009 by 3 of the most respected immunologists, and they argued, and they were talking about chronic viral infections, and in particular they study this persistent LCMV infection. And so here's a quote: "Ongoing immune responses hold these chronic viruses at bay while avoiding immunopathologic damage to persistently infected tissues." And of course, they've got this entirely backwards. It's the virus that holds the immune system at bay, or at least exhausts the immune system, so the virus can persist in order to be transmitted to the next host. And so that because they start with a premise that doesn't work, then they don't understand persistent viruses. How viruses manage to persist despite the impressive immune armamentarium of the host without causing overt disease is a great unsolved mystery. Okay, so there we are. And so what I want to do is I want to take a little bit of time to now talk about the idea of immune effectiveness and the difference between the adaptive immune system and the innate immune system, and whether that makes a difference. Now, having just heard the last talk, I'm going to have to rethink this because I never thought about the size of the host and the difference between host size and the effectiveness of the immune response. But I think density of hosts, in terms of coevolution with a pathogen, probably does play. I have thought about that a little bit. I also think it's a great idea to study this host differences in immune responses, but maybe the National Institute of Allergy and Infectious Diseases should fund this because it seems to be so relevant to things like West Nile disease and things like that. However, it's not easy to get grants there either, I can tell you.
Not looking for a free lunch.
There ain't no free lunch now, baby. Okay. So another point I like to make for immunologists is that the course of disease is determined not so much by the immune system, but by the pathogen's evolutionary history and its ability to overcome the immune system. So here's, I think, a very interesting example of this. So flaviviruses have a vector, like mosquitoes, and they actually infect mosquitoes and actually replicate in mosquitoes. And then they, of course, the mosquito bites the vertebrate, and then the vertebrate gets the infection. So this is one of the few pathogens or viruses that actually replicate both in insect cells and in vertebrate cells. And the interesting thing about this is that when you infect vertebrate cells with these flaviviruses, they suffer damage. Where if you infect mosquito cells, they are persistent non-cytopathic infections. The vertebrate flavivirus infections are associated with significant direct pathology and immunopathology, whereas the insect vectors sustain lifelong infections with little apparent cost. So here you have the vector with an innate immune system only, and the host with an innate plus an adaptive immune system, and all pathology is directed at the host with both immune systems. And the reason I would simplistically say is that, of course, it's to the advantage of these flaviviruses to do all of their replication and their pathology in the host and allow the vector to freely roam and infect the host. So it really is not the immune system that determines necessarily the severity of the infection or the disease process. It's all about the evolution of the pathogen. So the conundrum, which I've tried to say several times is that Natsatostomes have an acquired immune system and absolutely require it, whereas most species on Earth thrive without antibodies or T cells. And so I just want to quickly address this idea that invertebrates are short-lived and non-complex. And this is just for fun. Sea urchins essentially are ageless, and the age of sea urchins can be determined because in the 1960s we did a massive radio labeling experiment. It was called atmospheric testing of nuclear weapons. And so you can actually go in and test the age of these sea urchins, and they found that in certain places, I think it was Washington State, there was almost a 90% probability that a sea urchin would live to 100 years old. So these are long-lived animals. Here's arthropods, and you can see that within the same colony, that queens can live 10 or 20 years, whereas the workers live only a few years. So their longevity, of course, they have longevity. One of my favorites is the dung beetle, which a lifespan in the wild of about 2 years, lifespan in captivity about 5 years, about the same as our favorite model vertebrate, which is the mouse, and yet these guys spend their time rolling dung balls. Lobster, they have a very complex ecology and a very complex life pattern. They have communities. The males defend the females when they're molting. They do all kinds of complex things. So I don't know about complexity, but it seems like they have plenty. And they live 30 to 50 years in the wild. And their total mortality of juveniles and adults, way back before they were overfished and so forth, was only about 2%, a little more than 2% per annum. And that's including storm damage and predation and all sorts of things. So there's no evidence that an animal living a long time without an adaptive immune system succumbs to disease at any type and in any form that's at a higher rate than, let's say, mammals do. And this giant clams essentially are ageless and they can live 100, 200 years. And they're filtering the entire ocean through this poor climate. So, there's just no evidence, of course, that invertebrates are short-lived, and that's not an explanation for their ability to survive without an adaptive immune system. And finally, these are 4 species of birds, and this is their longevity profile, you might say. And what you can see is, and so this is the age and then the percentage of the population that lives to that age. And what you can see is that there aren't any outliers. Oops, there aren't any outliers. You never see a line like this where some of these guys live like this, and then there's a small portion of the population that lives forever. When you find long-lived animals, that means that that's their longevity curve. Okay, and that's a whole topic in itself. But so here's the enigma of the immune system. Morbidity and mortality rates vary widely in metazoans across all levels of organization. The forms of immunity vary, and yet there's no correlation between immunity and disease-related mortality. There's differences in mortality among species, and it comes and goes, and it's temporal, and there can be epidemics and infections. But as far as I can tell, reading these meta-analyses and reading the life histories of animals, there's no concrete correlation between those animals that have an adaptive immune system and those that don't. So the enigma is that individuals that are vertebrates that are lacking any component of their adaptive immune system then are highly susceptible to opportunistic infections. So if you haven't evolved an adaptive immune system, you don't seem to need it. If you have evolved an adaptive immune system, you absolutely need it. And so how do we explain that? Well, the way I like to think about it is that virulence correlates with mechanisms of transmission, and therefore I'm going to argue that the reason you need an adaptive immune system if you've evolved one is that your pathogens have evolved a virulence that takes that into account. And when you lose that immune system, they're over-virulent. All right. So, but I think this is a really neat correlation, and this was done by a guy named, this was, I think, at least popularized by a guy named Paul Ewald. And so he argues that there's a direct correlation between the mechanism of transmission and the level of virulence. So if you look at directly transmitted pathogens that require host mobility, they are largely benign. Rhinoviruses, or all the different viruses that cause a common cold. Whereas that diseases that have an intermediate vector in which they don't require host mobility can be much more virulent. And of course, these are the examples. Parasites that are enduring also don't depend upon the mobility of the host necessarily, and they can be highly virulent. So anthrax, of course, can last decades as spores, and so once an animal dies, it can sit there in the desert for 10 or 20 years, and it can still be infectious if a healthy host comes in contact. And the same is true for smallpox. Someone's actually done an experiment where they took a scab from a smallpox and put it on the lab bench, and 10 years later it still had the same virus titer. Smallpox viruses are incredibly stable. And then there's a fecal oral route. This doesn't require the mobility of the host either, and cholera is an example. And then secondary hosts is a whole discussion in itself, but they can have variable virulence depending upon the specific conditions. And finally, venereal diseases are always chronic, persistent infectious agents because there's just not enough sex in the world. If we had constant sex, then it could be an acute disease, I suppose, but that's just not the way it works. So all of the virulent diseases, are persistent diseases. So what you get from this kind of a correlation is that it's not the immune system that determines how virulent or how virulent a pathogen is. It's all about the background of the pathogen. And this was nicely written up by this fellow Paul Ewald. Okay, so a summary: pathogens carried out to carry out replication and transmission. If it kills the host before it's transmitted, it dies out. An overly virulent pathogen is a dead end. Ebola, for instance, in Africa, infects 29 people and then dies out until there's another reservoir infection of a human being. The more rapid the transmission, the more virulent the parasite. So that sort of gives rise to these crowd epidemic diseases. If you have a very dense population where the virus or the bacterium can be spread easily, then it can evolve to high virulence, whereas if it requires a long time to find the next host, it tends to be less virulent. Parasites evolve toward an optimum, and it's a trade-off between virulence and transmissibility, and virulence and persistence are highly correlated with the mode of transmission. So all of these things I'm bringing up is to argue that we, as immunologists, we need to think much more about the way diseases progress and how they evolve, and not put all the emphasis on understanding every last detail of the cytotoxic T-cell response in order to understand these kinds of questions. And this is so this is my sort of simple idea of host pathogen coevolution. The parasites target the most prevalent, the highest frequency host, and inevitably there's diversity within the population, and so these hosts might be slightly less sensitive to this parasite, and with time this can change, and so this host might become the most prevalent, and then there's more diversity, and on and on and on and on, ad nauseum, or ad infinitum rather. So it could be ad nauseum too. And so when you think about host pathogen interactions, it's sort of this red queen idea where you can run as fast as you can and you never get anywhere. And so Alice said the most curious part of the thing was that the trees and other things around them never changed places at all. However fast they went, they never seemed to pass anything. Now, I wonder if all the things move along with us, thought poor puzzled Alice. And the red queen says, now here you see it takes all the running you can do to stay in the same place. And that's what we as hosts are doing. We're running as fast as we can. We never get anywhere. And of course, this comes from a famous paper published by Lee Van Valen, in which he proposes in the great majority of cases, what one species gains, others lose to the same extent, and vice versa. So there's a constant coevolution, and in at least a two-component system, you never get anywhere. Okay, so my proposal is that the limitations to the virulence of an infectious agent are not caused by the immune system of the host, but rather it's intrinsically self-limiting. And I want to go over the concept in a little more detail. How long do I have?
You have an hour, so.
Oh, okay. So this is the way epidemiologists have looked at this, and this is limited. This kind of modeling is, in this particular case, is limited to a one-on-one interaction, but it involves this susceptible, infectious, and recovered model, and then you can throw in another part of this, so that some individuals will become immune, and so this would be the equivalent of vertebrates. So they're susceptible, they become infected. If they recover, they can become immune. And I'm arguing that this is largely restricted to these crowd epidemic diseases. In terms of all of the co-evolved diseases that I mentioned before, you never clear them. So alpha, beta, gamma, herpes viruses, and you never ever clear those viruses. And so they're talking about recovered and immune doesn't mean so much. But at least in terms of these crowd epidemic diseases, you can talk about this. For invertebrates, the idea is they're susceptible, infectious, recovered, and then they go back to being susceptible if, in fact, they're able to clear a disease. So this was worked out by Ray Anderson. I mean, he was one of the first to actually apply differential equations to try to model this. And I think it gives a really interesting result and some real-world predictions. So what he did is he just modeled this. He made X the susceptible population, Y the infected, and Z the immune. And then he wrote differential equations to try and understand how they varied with time. And so he put several components in: a birth rate, a death rate, and this death rate takes into account limitations of density in the population. And then he put a term in that was about infection. So it was the number of susceptible individuals times the number of infected individuals times a constant. So this is a highly simplifying element to this model, in that it proposes that the probability of being infected is simply a matter of the concentration of the susceptible and the concentration of the infectious individuals, kind of like a second-order chemical reaction. Now, and we know that the structure of communities is not like this, it's not perfectly mixing and so forth, but this is a simplifying part of this model. And then there is a component where individuals that were infected, immune, and then lost their immunity now became susceptible. So this is loss of memory. So this is the first equation. This just is an equation for the susceptible individuals. The infected individuals have the same terms. So it's the infected ones minus the death rate, and then there is mortality from disease. And so this is an important component of this model. And in addition, there's recovery from being infected. So this is the number of infected individuals. And then for the immune individuals, it's dependent upon the recovery. So recovery from infection, again the death rate, and then once again, and then minus the loss of memory. So when these guys lose memory, they're no longer immune, and they would become susceptible. And then here's the total population over time. And what they found for this type of analysis was perhaps intuitive at this point, but perhaps not. And that is, if you look at the population depression versus mortality from disease, so that's this component here, what you find is that there's an optimum in terms of the virulence, or the ability of a pathogen to cause population depression. And increasing virulence decreases that population depression. Moreover, if you look at the equilibrium infectivity, that is the number of infected individuals over the total number of individuals, with increasing mortality, the number of individuals in the population that are infected decreases. And these different curves here are just differences in population rate, and showing that that really doesn't have too much of an effect on the overall function of this. So what they concluded from this, well, first let me, I got ahead of myself. So then they actually did an experiment. They took a box of mice, and they either added 6 mice or they added 0.3 mice per day, and then they looked, and then they infected them with Pasteurella, and they looked at the population increase. And what's shown in the dots is the experimental data, and the solid lines are the model. And it's almost too good of a fit. It's hard to imagine that it worked out that well, but this is what they published back in the 1971. But interestingly, if you look at this constant, this mortality from disease term, this was their model, and this was their experimental result. And what you can see is that for Pasteurella, it looked like the mortality from disease constant mapped almost exactly the optimum for the ability to cause a decrease in the population. And, well, maybe that's just true for Pasteurella, but they did it for mousepox virus, ectromelia, and they got pretty much the same result. So what they conclude from this, in brief, highly pathogenic organisms are likely to cause their own extinction but not that of their host. If a total immune category of hosts is absent, recovered hosts passing directly back to the susceptible class, as may often be the case with microparasitic infections of arthropods, i.e. invertebrates without an adaptive immune response system, a pattern emerges which is very similar to figure 1. So there's an emergent property, this is my conclusion, of host-pathogen interactions, at least on a one-on-one basis, co-evolving basis, that is not particularly dependent on specifics and limits the pathogenicity of the parasite in a simple one-on-one interaction. So this is what I try to argue to immunologists, that when you think about this arms race that people talk about all the time, it's kind of it's not a fair fight. The pathogens always have the upper hand. They've seen that same immune response thousands and thousands of times in previous generations. They know exactly what to expect. They've worked out all the ways they can get around the immune system. That's why they're successful pathogens. So the idea that the immune system can combat that is true to some extent, but on an evolutionary scale, it isn't really. So virulence co-evolves with the immune system of the host. The more effective the immune system, the more virulence mechanisms evolve, but no more so. The immune system is an appendage, evolutionarily grown in response to millions of years of constant pressure by infectious agents. It's an appendage in the sense that it hasn't really increased your insusceptibility to infection. And so what I would argue is that on an evolutionary scale, the immune system does not prevent parasitism. Now, this was another little, there's some other things I wanted to talk about, but I think I'm going to let it go for now because we've been through this long enough. So what does the immune system do? I think it prevents infection by most microbial agents that are not adapted to parasitism. So it requires that an infectious agent specifically adapt to a lifestyle of parasitism. And in many cases, it requires that an infectious agent specifically adapt to a host. Now, of course, Borrelia, for instance, is adapted to many different hosts. So that it breaks down, especially with bacterial infections. But for viral infections, it's pretty closely correlated. That is, viruses tend to have a limited number of hosts. There are exceptions, of course. But in the real world, they have many, they're often limited by their hosts. It makes transmission more difficult. There are higher numbers of organisms required. It may manage mucosal flora, and it may have a role in cancer surveillance. I think that's a weak one. I think the immune system really is evolutionarily selected for infectious disease, but people often talk about cancer surveillance as well. It does not seem to prevent disease in an evolutionarily or ecological sense. And then I just want to end with one of my favorite little ditties here. Great fleas have little fleas upon their backs to bite them, and little fleas have lesser fleas, and so on ad infinitum. And the great fleas themselves, in turn, have greater fleas to go on, while these again have greater still, and and so on. This actually came from Jonathan Swift, a simpler little ditty. Okay, and this is La Jolla. This is the view out of my office, actually. And I'm very pleased to be here. Thank you for having me. Yes.
So you talked a lot about one-on-one interactions between hosts and parasites. If you accept that most animals are co-infected with multiple parasite taxons, what role do you think co-infection plays in selection on the immune system?
Yeah, so that's the question was, I was really talking about a very simple case for the one-on-one infection with a host and a pathogen, whereas that's not the real world. Hosts have many pathogens, and pathogens can even have multiple hosts, species. So how would this play out if you talked about a real-world situation where the hosts have many different pathogens? And simultaneously. Yeah, I mean, thinking about this seems, for me, is hard enough on a one-on-one basis. I really, it's just way too complex. I haven't been able to think about that much at all.
Can I throw out a follow-up that maybe even adding to that complexity for you or others to comment on? And that is, the sort of conceptual framework you're putting out here is this idea of this co-evolution over a very long period of time, but some of the infectious diseases that we're thinking about the most these days are emerging diseases where the vast majority of which have jumped to humans from some non-human animal. And I wonder if you have thought about the immune system and this co-evolutionary model within the context of these jumps from other species. There's obviously a host range involved here.
Yeah, so I can state the obvious, and that is that when a pathogen jumps from one host where it's highly co-evolved to another, you can have almost any level of virulence. You can have nothing, which is probably the vast majority of cases, and you can go all the way up and have lethality, like Ebola, that just burns itself out. And it's just unpredictable, whether it's SARS or Ebola or HIV. HIV turned out to be the unfortunate case in which it was highly virulent, but only after a long period of time, so it was still contagious. And it's just not predictable. You just don't know what the virulence is going to be because it's unselected. I mean, I don't know anything more than that. Anybody else? Anything else to say? Yeah.
I had a question about what you mentioned about the all of the vaccines that have been successful, or most of them anyway, have been for these acute crowd-borne diseases and not so much for the co-evolved diseases. Do you think that that's a matter of resources that we've put into the development of those vaccines, or do you think that there's something fundamental about a long co-evolutionary history that makes it much more difficult to develop an effective vaccine?
I think it's the latter, but I don't really have a good conceptual way of understanding that. I mean, clearly, those diseases have many mechanisms to overcome the immune system, but maybe not if you had a pre-existing immune response to those pathogens. So, for instance, if you had an effective anti-HIV immune response before you were infected, and so you could neutralize the virus, for instance, maybe it would never have a chance to infect the individual. But there must be a correlation, because it's not just for lack of resources. Up until last year, we were putting $500 million a year into an HIV vaccine and got nothing. And malaria is, for other reasons, malaria is also very difficult. It's such a complex life, cycle, and it just seems to be insusceptible not susceptible to immune responses. So I think it's the latter, but I don't know for sure. And just to be clear, there is, the one that we've gotten is against human papillomavirus, which is a long co-evolved disease, but there are very few other examples. Everything else are these crowd epidemic diseases where we get fabulous immunity, and vaccines work really well.
Another question for you, and it comes from my desire, as many of us in this room do, to have tools to ask animals about their immune competence. You talk about, a population of humans or animals that all have sort of not cleared a virus. I'm always interested in any particular species in finding out if there's some virus that most of them have been exposed to, and then wondering if I can use something like viral load amongst those animals to actually say something about variants in immune competence. Your thoughts on that?
So say that again.
So, if you have a population that is either co-evolved or recently experienced some sort of viral infection, and most of those, your chances are not really clearing that virus.
Okay.
Can we then use something like viral load or interferon levels or something to as a tool to say something about the relative strength of particular animals' immune responses? Do you think if it's persisting, and you've got 90% of the animals in the population that have a virus, can we use that as a tool to be able to say something about which animals have stronger immune systems?
Well, the first thing I would say is, and this is the part I left out, is that you take these, if you look at the persisting infectious agents, you put them in two categories. One is persistent and one is latent. And they give a very different result to the immune system. So persistent virus, you're left with these T cells that are almost completely anergic and unable to respond. They don't have any of the characteristics of immune memory. And the example is these arenaviruses in mice. On the other hand, if you look at a latent virus like herpes, the T cells that are there have all the characteristics of a memory T cell. They're highly responsive, and you can transfer them to another host and get a great response. And so that's the first distinction to make. Those are two very different types of immune response. One, you might be able to get some information on the herpes virus because the T cells are so, but you never clear them. So that's 100% infectious, 100% non-clear in human beings at least. And I think it's true for mouse cytomegalovirus as well.
But if you could quantify those antibodies, does that mean anything? Right? So could I develop a quantitative measure, basically taking advantage of the fact that these animals have all had herpes viruses?
Mm-hmm.
Can I use some sort of quantitative measure, antibody levels or T cells or something, to say something about which animals seem to be pushing that infection down better than others?
Yeah, I think so. If you looked at the ability of the T cell to respond, you'd have to correlate it to the recrudescence of the virus and see whether there was some correlation. But yeah, I would think so. Has anyone studied, for instance, shingles is now a big deal in people of a certain age. And it's a very painful disease, and it's probably the immunopathology that causes such pain. And so is there a correlation between your T cells or your antibodies and whether or not this virus reemerges? And that could be a clinical study that someone could do. I'm not sure it's been done.
I'll have a comment that might be pertinent to your question in the earlier one, which is the parasites where vaccines fail, many of those taxa possess mechanisms for antigenic variation.
Yeah.
So that actually is what, because they have that, they
Yeah.
have gene duplication or shifting of antigens within one strain, that's going to govern whether or not the vaccine works. And for parasites that have that, it would make what you're describing problematic. Because even though it looks like the same species of parasite, it can antigenically shift. So wherever you're looking at in the time course, you can influence whether the antibodies present are doing anything for resistance.
Yeah, it's famous for panosomes, but I think lots of organisms can.
Plasmodium,
Yeah.
Even the plasma, the bacterial pathogens have that. Lots of these parasites have got methods of antigenic variation.
But if you were looking for parasite burden instead of an immune response, would that be a measure of immune fitness of the host?
Only, well, that then leads to the question of resistance versus tolerance, and whether or not the parasite's actually doing anything bad to host fitness, which is a whole other set of problems. So I don't know that parasite burden would actually be a good metric, would be the best metric for immunocompetence. I think it's one way, an important way to look at it, but I don't think it's the only one.
I think a lot about all of this, and I really issues, and I think when you measure, right, you want to know what the titer of antibodies is. You think the antibodies are breathing, and you care about it because your animal's gone through safety. You can figure out the relationships among the immune response, the parasite, and the virus, which of course is the holy grail, but to the extent.
But that's a good point. Lots of antibody responses are non-neutralizing for the viruses. So how does that factor in?
You can have a huge antibody response.
I mean, you measure HIV infectivity because you have antibodies. That's typically the way you measure it, and they don't do anything. Probably because of antigenic variation, but not necessarily.